Thank you. Thank you for that introduction. Today, I'll be talking about considerations on in vitro release testing for long-acting products from quality control perspective. Before I begin, let me remind you, the presentation reflects my personal views and should not be construed to represent FDA's views of the policies. So in today's talk, I'll talk about in vitro release test. The current approach that we are using as we are looking at these in vitro release specifications, and I'll touch upon the product-specific in vitro release test, discriminating ability, and setting acceptance criteria. Some of the most common deficiencies that we see and recommendations related to that. Then I'd like to share some thoughts about where we are moving forward, and then I'd like to leave you with some take-home message at the end of the presentation. Okay, now I know everybody has seen these, however, I have to say it, so here it goes. A quality product of any kind, a car, a cell phone, a laptop, consistently meets expectations of the user, and drugs are no different. Patients expect safe and effective medicine with every dose they take, and pharmaceutical quality is assuring that every dose is safe, effective, free of contamination and defects. And this is what gives patients confidence in the next dose of medicine. With that, let me get to the specific topic of the day, in vitro release specifications. Now, there are two parts to in vitro release specifications. One is about the actual in vitro release method and the acceptance criteria. From quality perspective, the purpose of in vitro release specifications is to detect variations during routine product manufacturing, detect changes during product storage that may negatively impact product performance, and it can also be used to support minor or moderate CMC changes. There are three ways to arrive at an in vitro release method. If there is a method published in USP monograph or available in FDA database, explore that method and then ask, is this method appropriate for my product? Is it appropriate for this product under consideration? Now, things to evaluate as we are doing this: is the method robust yet simple enough for routine testing? Is it discriminating across trends? Can it discriminate for changes, the meaningful changes in critical quality attributes? Can it reject non-BE batches? Now, after considering these questions, if it is concluded that the method is appropriate for the product under consideration, go ahead, you can use that USP or FDA database method. However, the answer is no, then develop an appropriate product-specific method. If there is no method available in USP monograph or FDA database, develop an in-house method. But there is an option. There is an option to develop an in-house method that is appropriate for your product, the product under consideration. Now, we're talking about complex products. So what are some of the challenges? So some of the challenges from the in vitro release method perspective. These are complex products and complex excipients. So even small changes in excipients or manufacturing process can lead to significant changes in product release. And therefore, it is important to understand and identify the critical material attributes, the critical process parameters, and critical formulation variables. All these critical factors, they can affect product performance. And thinking in the same lines, even small changes in these can lead to changes in product performance. And we need to know this information, not only during product development, but also throughout product life cycle, as it may go through certain changes. And it is not practical to conduct in vivo studies to understand this information. However, if we can develop an in vitro release method that can capture this information, that can tell us when there are changes in these critical attributes that is specific for this product, it can be a great tool in ensuring product quality and in turn ensuring optimum in vivo performance. So I've been talking about the product specific method. How do we do that? Well, here are some points to consider. The three main points when you're developing a product specific method: identification of appropriate testing conditions, discriminating ability, and selection of appropriate acceptance criteria. As we are going through the process, it's important to consider the CMAs, the CPPs, the CFVs, and how these affect in vitro release and product performance. So let's first look at some of the factors to consider when identifying testing conditions. Whenever there is a release involved, whenever there is a release step involved, in vitro release test is a critical quality attribute. So as we are developing, as we are identifying the appropriate testing conditions, know your API, know the solubility of the API. Is it pH dependent? Is solubility not changing throughout the physiological pH range? Know this information. It can help us in the next steps as we are developing the method. The next step is here outlined as selection of apparatus. Whatever apparatus is selected, generate data to identify the right flow rate or the right rotation speed for that apparatus. Now, because we are talking about complex products, sometimes non-compendial apparatus are used. So when the non-compendial apparatus is used for an in vitro release test, provide information about how the product is introduced or retained inside these apparatus. If the test goes on for a long time, steps should be taken to ensure that there is no evaporation from this apparatus. And of course, adequate validation. It's also helpful to provide dimensions of this apparatus in the submission, just so it's easier for us to understand as we are reviewing the method. In vitro release media. Now, sink conditions are usually recommended, and if that requires use of surfactants, evaluate different surfactants, different concentrations, so we know why this particular surfactant is used, why at this particular concentration. And of course, method validation. Method should be adequately validated. Since we are talking about long-acting products, here are some additional considerations. Real-time testing. Consider evaluating in vitro release through the intended period of product use to gain mechanistic understanding of drug release. Now, in cases where real-time testing cannot be used as a regulatory tool, as a regulatory QC method, you can develop an appropriate accelerated short-term method. Such a short-term method should be supported by appropriate cross-validation, demonstrating interchangeability of the proposed test as a quality control tool. Discriminating ability. I've been saying this word probably many times. So what is it? A discriminating in vitro release method can differentiate drug product batches manufactured under target conditions from batches manufactured with meaningful variations for the most relevant manufacturing variables. Meaningful variations in this context means usually 10 to 20% change to specified values or ranges of the critical variables. Use all the available CMC data, in vitro release information, in vivo PK data as a discriminating method is developed. Selection of appropriate acceptance criteria can also help in improving discriminating ability. That brings us to the next point, setting acceptance criteria. Acceptance criteria is based on data obtained for the proposed product, the pivotal clinical or PK drug product batches or bio batches and exhibit batches. Data from 12 units should be submitted. Consider all the available data from pilot batches, any failed or acceptable BE studies as you decide on the acceptance criteria. Because we are focusing on the long-acting products, consider setting 3-point specification that covers the early, middle, and late stages of the release. Now, as you're setting the ranges for these acceptance criteria, the general practice is to have a mean target value plus minus 10%, and then the last time point should be where 80% of the release occurs. If 80% release does not occur for a particular method, then the last time point should be when the plateau of release profile has been reached. So let me share with you one of the examples where in vitro release method was optimized. Now this is an example for an injectable suspension where the active ingredient was a low solubility API. Here is the data obtained using USP apparatus 4. We can see 3 profiles here. The blue one was obtained using the highest of the tested flow rates. All of the drug is released in first few minutes. So it is such a method cannot detect meaningful changes in product quality. So the method was further optimized with a lower flow rate, and we see a yellow profile represented here. Now this is a little slower than the blue. However, optimizing the method further gave us the green profile. Now we can see that the release is fairly slowed down, and such a method can possibly detect meaningful changes in product quality. But did it? Let's see. So as I mentioned, this is a suspension with low solubility API. So particle size distribution is a critical factor for these products. So using this new optimized flow rate for this particular method, data from 2 batches was submitted. The green one is the target batch with optimum particle size distribution, and the red one is an intentionally changed batch where the particle size distribution was higher than optimum. And this method, as we can see, can clearly detect the changes in this particular aspect. Method validation. Now these are complex products, and there can be several sources of variability. And sometimes it's not very easy to identify where the variation is coming from. So as the method is validated, pay attention to the factors of the method that can have influence on in vitro release. So in this particular example, this is again for a complex injectable product. The red dots indicate release obtained at the optimum pH of 6.5. And as we can see, pH change from 6.5 to 6.3 or 6.7 gave rise to a large variation at that 2 and 4 hour time point. Similarly, temperature can have a similar effect. Optimum temperature represented here with the red dots, and 1 degree higher and 1 degree lower. Now we can see that it's giving rise to a large variability at these 2 and 4 hour time points. So understand this information as method is being developed, and whenever we see such a trend, it's important to take precautions to control these method parameters. That brings us to the next topic, the common deficiencies and recommendations related to that. Method development and validation report. Now, for a deficiency about method development, what we would like to see is data demonstrating that whatever is the selected method, it's appropriate for the proposed product, and validation of that in vitro release method. Demonstration of discriminating ability. What I would like to see in this is identifying the critical manufacturing variables and data obtained from batches with intentional meaningful variations to these variables. You can also conduct statistical testing comparing the in vitro release profile of the changed versus the target batch. And the third most common deficiency is about appropriate acceptance criteria. So acceptance criteria should be based on data obtained from the proposed product. And whenever there is more than one time point specification, consider using ranges rather than open-ended not less than or not more than type of a time point. So here is what we have been doing so far, and I would like to take the next few minutes to share my thoughts about where we are moving forward. We are moving forward with clinically relevant drug product specifications. These are those specifications that ensure consistent product performance for the marketed product relative to those achieved by the clinical trial formulation. Goal of an in vitro release test should not be just to pass or fail a test, but it should rather be develop drug product specifications that will ensure adequate performance of future batches prepared within the limits of acceptable in vitro release specifications. And to do that, it's important to understand impact of changes in critical quality attributes on in vitro release and systemic exposure to establish what we call safe space. So safe space is the space within which the proposed in vitro release specifications are demonstrated to be biopredictive or clinically relevant. And how we can establish safe space? Well, here are three ways to get there. The first one, based on pilot and pivotal BE batches. Identify extremes of dissolution profiles for products that are found to be bioequivalent to one another and the RLD. So the future batches with the profiles between these extremes can be considered bioequivalent. Safe space can also be established based on the in vitro in vivo correlation established via conventional approaches, or safe space can be established via in vitro in vivo relations, the IVIVR or IVIVC, established via physiologically based biopharmaceutics modeling. Now here is an illustration that can help us understand the concept. The blue, red, and green profiles, they represent the batches that are bioequivalent. And so they represent the safe space. The black profile, which is outside of this safe space, and therefore we do not know how this particular batch will perform in vivo. So this has been a long day and last presentation of the day. So even if you couldn't pay attention so far, here is the take-home message. I want to emphasize the importance of product-specific method. Importance of submitting data demonstrating that the selected method is appropriate for the product. Identifying the CMAs, CPPs, and CFVs. Demonstration of discriminating ability. Data-driven acceptance criteria. Acceptance criteria should be based on the observed data to ensure should be based on observed data and never lose sight of clinical relevance. With that, I would like to thank my colleagues, Dr. Sandra Saras, Okpo Iradri, Om Anand, Kimberly Rains, Paul Seo, and my colleagues in Division of Biopharmaceutics. And thank you all.